Chapter 4, Parasitic Protozoa, Form, Function, and Classification. Form and Function, Part 4. When you look at the reproduction of these protozoa, you're going to see both sexual and asexual mechanisms. Remember, asexual means one cell splits, becomes two. Two genetically identical daughter cells. Okay? Genetically identical. Those two become 4, can become 8, 16, 32, okay? It just keeps going on and on. However many daughter cells, after how many generations, how many divisions, they're all still exact copies. Sexual reproduction is about increasing genetic variability. So it's still two cells, because remember we're talking unicellular things, coming together, fusing. Either the cells, as we're going to see, can completely fuse, mix chromosomes, and then split. Two genetically unique daughter cells, or the cells could fuse just part, mix nuclear contents, and then divide. End up with two genetically unique daughter cells. So the sexual reproduction isn't about intercourse, as we see with animals, the mating in that form or function. It's about somehow mixing genetic materials. So asexual reproduction, let's start with this one. Asexual reproduction, remember, one cell splits, becomes two clones. You can do this with binary fission. This is literally binary, means two, fission, splitting, one cell divides, two completely identical resulting daughter cells. This can be done, what is it referred to, symmetrogenic. Got to remember, some of our protists are going to have an anterior end and a posterior end. And off the posterior end will be some flagella. So, can't divide, willy-nilly, because one of the daughter cells will get flagella, the other won't. So what they'll do is they'll not only divide the nuclear contents, but they will also double the number of flagella at the posterior end, and then divide down the center. So when the cells split, this cell has a flagella, this one also. So it means they have to go through and the kinetosomes, the dictosomes, the mastodon system, if it has one, all of that has to be doubled, has to be copied, and then equally split during the cytokinesis, the cellular division. If it's a ciliate, like Tetrahymena, then doesn't have to worry about where. So the posterior end, the anterior end, and it's just going to divide somewhere in the middle, two resulting cells that will then increase in size, and then, away they go. One of the things you have to watch is how is the nucleus? How is the nucleus and the nuclear contents divided? Okay, we're going to have the mitosis you've learned about. Prometa, meta, ana, telophase. Okay, there may be some modifications to it. What do I mean by that? Well, for some of these protists, the nuclear envelope doesn't disappear like it would in our prometa and, prophase, prometaphase. Instead of the nuclear envelope, disappearing, it may still be there, and you still have the centrosomes, but they're now encased within the nuclear envelope that's just gotten bigger. Or instead of the centrosomes, remember those are the anchor points that anaphase uses to pull apart. Instead of that, you will have microtubules that span the nucleus. And that's what the motor proteins will just move along, pulling the chromosomes apart on. Instead of that, you could have, the microtubules just spanning the entire, diameter of the cell. And then the nuclear contents just get split by motor proteins in the nucleus, and then the nuclear membrane divides, and then the rest of the cell can divide sometime later. You can end up with multiple nuclear divisions still within one cell. Okay? So you have instead of, one mitotic event followed by cytokinesis. After telophase, then you get the dividing again, and the dividing again, and the dividing again. So in one cell, you could have, 6, 8 daughter nuclei. And that at some point later, that cell will then divide up, and then boom, you have 8 genetically identical daughter cells. Depending upon the genus species, if it's following this pattern, they could be referred to as schizogony, merogony, schizogony. The resulting cells, the 4, the 8, whatever it is, are going to be referred to as meronts, schizonts, merozoites, depending upon genus species and which stage in the life cycle this is occurring in. You see down there at the bottom that figure right there showing one large cell, and that's the host cell, and inside of that lighter area, you see, 3, 6, 8, 9 nuclei. There's probably more that just aren't in that depth of field. So at some point in time, that parasitic protozoa will go through cytokinesis. But instead of one cell becoming 2, each with its own nuclei, what you're going to end up with is a protracted cytokinesis that's going to result in 8, 9 different cells all at once. So when you look at it here a little more close up, you can see that this multiple fission event is not going to be one cell divides and then that cell divides and those cells divide, giving you 8 cells of, over time. You're going to get nuclear division within one cell, one protocell. You'll end up with 8 or more nuclei, and then all at once you will get the cytokinesis event, and you'll have all the cells at the same time. Not 2, then 4, then 8, then 16 like you would with mitosis in our bodies. The gametogeny, the sporogeny, this is, the gametogeny is the making of the gametes, the multiple, divisions to result in cells that will be able to fuse. They now have the internal structures, the nuclei are ready to, and all that stuff so that they can fuse. You get mixing of the genetic material followed by mitotic events so that you end up with multiple daughter cells that are all still the same species but genetically unique. You also see what's referred to as budding. One large cell will get a little protrusion off the side that now has its own nuclei, may have some of its own organelles, and then it will wall it off. So you now have the large original mother cell and a bud or a daughter cell that's like a mini-me attached to it. That mini-me can break off, float away, and become its own thing, or it can just get bigger, 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 and they stay attached. Still, they may be attached, but they are still every cell for itself. You can also get, as you see here, what is referred to as endodigogyny. I hate these words. Endo means within. So you have the fission still occurring within. So how is this different than what we saw before with the schizogony? The schizogony is nuclear division, then cytokinesis all at once. Here, the resulting daughter cells are still encased. The cytokinesis has occurred, but these are daughter cells still encased within the cytoplasm of the mother cell. The mother cell at some point in time will die, burst open, and release the daughter cells. But in here, the daughter cells are independent. They are metabolically active, just housed within the mother for a time. And when you look at the gametogeny, the gametes, gamonts, these guys, depending upon whether you're talking amphiphymetic or automitic, meaning it makes the gametes that it can then fuse with on its own, makes both that will then fuse, or you need two different organisms making gametes that will later come and fuse together. Pros and cons of each. If it's amphiphymetic, meaning you have to have two organisms that will make gametes in their lineage, and then those lineages fuses, and you get resulting genetically unique. Versus the automitic, means it can make its own gametes, and then those gametes may fuse together. Think yeast cells. Yeast cells are going to be amphiphymetic. Think back to intracell and molecular, maybe genetics if you've had it. The A or the alpha, okay, have to make mating factors. A can only fuse with alpha. Alpha can only fuse with A. Two alphas can't do it. Two A's can't do it. So this is a means of increasing genetic variability, genetic viability in the resulting generations, versus just making sure that there is another generation. That's what the automitic does. It makes sure that there's going to be more, because you can't always run into another parasite. If you're a gastrointestinal parasite, the chances of you running into something that is, an alpha and you're an A, well, you're in somebody's gastrointestinal tract. If you run into another protist, well, you're just lucky enough to have done that. We also have what is referred to as conjugation. Conjugation is when two cells fuse, but not completely. They fuse, get an opening between their plasma membranes, and then they swap nuclei. Swap nuclei, swap nuclear material. So that way, once, you get the fusion of the nuclei, mixing of the genetic material, those nuclei divide, and you're off to the races. We see this in protists that have two nuclei already in them, a macro and a micro. Macronuclei, by their name, are bigger, micro, smaller. They both contain the same chromosomes. The macro controls certain metabolic functions, micro controls others. Before conjugation can occur, the macronuclei basically just disintegrates, gets broken down. The DNA gets broken down, all of it. Cells fuse. Micronuclei double. They reproduce. They make copies of themselves, and then they swap over micronuclei. Micronuclei from one cell fuses with the one from the original. They become macronuclei. Go through, rounds of mitosis after that within, but no cytokinesis. So you get multiple micronuclei, multiple macronuclei, then you'll get cytokinesis, and now you have resulting genetically unique daughter cells. And you see here in this picture here, the cells have fused, the micronuclei are increasing in number. They are going, through their own little copying phase so that they can now start swapping them over. They will fuse, become the macronuclei, and you have genetically variable daughter cells.